Okay. Good afternoon, friends. இன்னைக்கு நம்ம பார்க்க போறது cell division, cell cycle and cell division. Okay. Cell cycle and cell divisions. Okay. Cell division and cell cycle and cell division. Okay. It main are very important for growth. Okay. The cell cycle and cell division are very important for growth and reproduction. அதனுடைய வளர்ச்சிக்கும் அதனுடைய இனப்பெருக்கத்துக்கும் இந்த செல் division is very important, cell பிரிதல். Okay, so what it happens, every single cell it divide into two, okay, or divide into multiple form. Single cell it divide into two or more than a two, it is called divisions. Okay, divide into division, or we call this fission. So growth for the cell and the reproduction of the cell is just helpful for this cell division. Okay, so this is a case. First is it omnis, okay, e cellula cellulae. Okay, omnis e cellula cellulae. Okay, this is a concept it is given by Virchow. Okay, Virchow he said that all the cell it is developed from this pre-existing one. Okay, so cell division, the concept it comes from முதல்ல யார்ட்டந்து வந்துச்சுன்னா இந்த omnis cellula cellulae, Virchow it is concept it came. ஏற்கனவே இருந்த செல்லுன்னா ஒரு செல் வளர முடியும் அப்படிச்சு. So the cell divisions mostly divide into three categories. Okay, it's a cell division. Okay, one is A mitosis, okay, and mitosis and meiosis. Okay, so the cell division further divide into three category: mitosis, amitosis, mitosis and meiosis. And this cell divisions, okay, further it is, when you come to this two category, one is this non-dividing stage, non-dividing stage, another one is dividing stage. Okay, so non-dividing stage we call as G0, okay, and dividing stage is further is divided into G1 and S stage and G2 stage. Okay, so it is called as interphase. Interphase. Okay, so cell division is further divided into so many stages. Okay, amitosis, and amitosis it is mostly it is happens single cell. Okay, single cell organisms it does happens. So mitosis and meiosis it is multicellular organism. Okay, and here what you want to know that, okay, so what kind of process it happens in mitosis and meiosis and amitosis, but mainly and the process they have, okay, the mitosis further divided into 5 category: prophase, metaphase, anaphase and telophase. And meiosis and further divided into meiosis 1 and meiosis 2. Meiosis 1 it is further divided into prophase 1, prophase 2, and metaphase 1, metaphase 2, anaphase 1, anaphase 2, telophase 1, telophase 2. Like we have a different process which is occur in the meiosis. Okay, so each process is very important. It's a very simple unit. It is, you can easily understand, but the question is come from in which stage and what process it has happened. Okay, they are going to ask. Okay, the first, the cell cycle. Okay, cell cycle, the process discovered by cell cycle, the process discovered by, okay, Harvard. Okay, cell cycle that discovered by Harvard and Pilk. Okay, Harvard and Pilk. Okay, in the year 1913. Okay, so 1953 he discovered it. Who? So cell cycle, the process discovered by Harvard and Pilk in the year 1953. So it is a continuous, okay, it is a continuous chain like, okay, process to divide one after another. Okay, another we called as, it is a sequential process. It's a sequential process which is occur in the living organisms. So cell cycle that discovered by Harvard and Bell in the year 1953. So the process which is occur in the living organism, it is a continuous chain-like process. They divide one after another, or it is also called sequential. It is one after another sequence. It is occur in the living organisms. Okay, so it is a sequential process, divide and redivide. It has happened in the living organisms. Okay, now we move to this. I already said it, interphase. Okay, interphase, it is interphase. It is divided into G1. Okay, sorry, G1, S and G2 phase. Okay, G1 phase or gap phase, S phase, or it is we called as synthetic phase, G2 or G2K phase. Okay, G gap phase, or it is called as synthetic phase. Okay, G2 gap phase. Okay, so this kinds of phase which is occur in the interphase. So interphase, mainly the first what it happens, they have a process interphase, they have a growth it is, okay, the growth it is occur in the G1 phase. First thing is growth is, and second what has happened, they have a differentiation. You can able to differentiate which is and what. Okay, so this G1 phase, mainly the process which is occur in the growth. Okay, வளர்ச்சி இருக்குது. And the second thing is you need to differentiate which organism which part that we can differentiate. And second thing is synthetic phase. It is mainly DNA replications. The DNA replication, the process mainly occur in S phase. Okay, so synthesis of DNA, replication of DNA. Okay, synthesis and replication of DNA which is occur in S phase. Okay, and again, and there's another thing is chromosome also replicated. Okay, so first process G1, it is a growth, it is occur, differentiation is occur, and S phase, synthetic phase, the chromosome are replicated and DNA replicated and synthesis also. And again, and G2 mainly, again it happen, it is again it is when you come to this G0, there is non-dividing. Okay, there is no more dividing phase, non-dividing phase. Only it is this things, okay, increase the size. Okay, so these are the things that happen in the things, okay, the growth and in this one. Next we move to this. Okay, so cell divisions. So I said it is cell cycle, it is a continuous sequential process, it is repeated process. Okay, repeatedly they divide and redivide. And cell division, it is also called as complex and also continuous process. Okay, so it is also complex and continuous process. Mainly due to cell division, we have a growth and we have a development also. Okay, for that reason, okay, it's a complex process and also continuous process. Due to cell division, it may occur in the growth. It is the cell, it is increase in size and length and breadth, and also they have a development which is occur in the cell divisions. Okay, so cell division, it's further, it is divided into three categories. A, mitosis, okay, mitosis and meiosis. Okay, meiosis further divided into meiosis 1 and meiosis 2. Okay, so meiosis 1 and meiosis 2. So this is the process which is occur in cell divisions. Okay, these are the types of cell division: mitosis, and meiosis. So cell division, it's a complex one, continuous one. Due to cell division, they have a growth and development. It's cell division further, it is divided into three categories, or they have three types: mitosis, and meiosis 1. Okay, mitosis, that is it discovered by mitosis, it is discovered by Remak. Okay, Remark, he discovered in RBC cells, red blood corpuscles. Remark, okay, he discovered this A mitosis, he discovered in this red blood corpuscles, okay, and also he discovered in bacteria. Okay, so RBC and bacteria, so okay, of the cells, okay, and mainly here what does happen? They have elongations, okay, of cells. It is occurred. So where? In the A mitosis, okay, in the bacteria, RBC, and this elongation. Who discovered? And Remark. And this mitosis, okay, mitosis, it is discovered by Becker. Okay, Becker. The mitosis is discovered by, okay, Becker. So cell division, it is a complex continuous process. Due to this, they have a growth and development. They have a three process: mitosis, meiosis, and amitosis. Amitosis is discovered by Remark, is discovered in bacteria and RBC cells. Mainly the elongation occur. The mitosis, it is discovered by Becker. Okay, another meiosis, it is discovered by, okay, meiosis mainly discovered by, it is Farmer and Murray. Okay, Farmer and Murray, it is discovered. So okay, Becker, it is discovered, okay, in the plant, okay, it's discovered in plant. In animal cell, he discovered in plumbing. Okay, animal cell, he discovered in plumbing. Okay, so cell divisions, A mitosis is discovered by Remark, and Becker discovered in plants, plumbing discovered in animal, Farmer and Murray discovered in meiosis process. Okay. Now we are going to see what is mitosis and what is meiosis and what are the process which is occur in mitosis and meiosis. Okay, you need to know, okay, first thing is what process and where, in which stage it is occur. Okay, so process, which stage is occur. Okay, the mitosis, already I said it, mitosis is discovered by Becker and plumbing. So Becker is discovered in plant and plumbing discovered in animal. So, okay, so mainly it is helpful for cellular reproduction. Okay, cellular reproductions. Okay, the cell multiply into, for the growth and development, it is helpful, this mitosis. Okay, so mitosis, mainly each cell, okay, each cell divide into, okay, two daughter cells. Each cell is divided into two daughter cells, okay, or it generates into two daughter cells. So they have a 4 to 5 processes. One is prophase, metaphase, anaphase, telophase, and cytokinesis. Okay. Further, the cytokinesis, okay, which is also divided into two categories. One is cell plate, another one is cell furrow. Okay, cell plate method and cell furrow method. So cell plate, it is common to plants. This is common to animal. So cell furrow method, it is occurring in animal cells. Okay, so prophase, metaphase, anaphase, telophase, cytokinesis. These are the 5 stages. Okay, it just occur in this stages. Stage 1, prophase. Thus, when you want to remember, okay, this stage is, I will tell you very simply. Or the propose பண்ணனான மென்டலான ஆன்டி ஒரு டீல் கொடுத்தாங்க, ஆன்டி வந்தாங்க, ஒரு டீல் கொடுத்தாங்க, அவன் சைட்டாவும் வரான். ப்ரொப்போஸ் பண்ணாமல் பொண்ணுக்கு, அதனால மென்டலான மெட்டா, அதனால அண்ணா ஆன்டி வந்தாங்க, டீல் கொடுத்தாங்க, சைட்டோ வரான். ஓகே, ப்ரோ மெட்டா அண்ணா டீலோ சைட்டோ. okay, propose பண்ணா, okay, profess, meta, mental ana auntie vandanga, deal கொடுத்தாங்க, cytokinesis. So this 5 process you are easy can remember. Okay, இத நீங்க ஈஸியா ஞாபகம் வச்சுக்கலாம். இந்த மாதிரி நீங்க நிறைய சின்னதா உங்களுக்கு தெரிஞ்ச வேர்ட்ல நீங்க ஈஸியான எளிமையான முறையில கத்துக்கலாம். Okay, prophase, metaphase, anaphase and telophase and cytokinesis. Okay, so this it is 5 stages which is occur in mitosis. நம்ம இதில் என்ன தெரிய போறோம்னா what we are going to see that in which process what it happens, என்ன நடக்குது, what it happen அப்படிங்கறத பார்க்க போறோம். So cell plate method it is in cytokinesis, it is occur in plant, cell furrow which is occur in animal cell. Okay, so now we are going to see what it happen in prophase. Okay, prophase stage la என்ன important process நடக்குது. Okay, in the prophase stage, okay, when you come to this prophase, it is a short stage. Okay, it is a short, very short, the stage is very short. Okay, what it does happen, and also in the start and also a thickening stage, cell it become thickening. Okay, thickening stage, short and thickening, okay, of chromosomes. Mainly, so this process is a start stage, and so the chromosome it's going to thickening, okay, in this prophase. And okay, and the second is the metaphase. Metaphase, what it happen, equatorial, okay, plate, okay, equatorial plate it happened. Okay, here metaphase. Another thing is disappearance of what happened, nucleolus and nuclear membrane. Okay, so what it happened, prophase, the stage it is short stage, it thickening of chromosomes, and metaphase it is a equator plate and disappearance of nucleus and nucleolar membrane. And another thing is, okay, here and one more thing is it happen, formation of spindle. Okay, formation of spindle formation, it's occur in this metaphase. Spindle formation it occur in metaphase. Okay, so short stage thickening of chromosome prophase, metaphase it is equator place and disappearance of things. Okay, and another thing is anaphase, what will happens, that is split two daughter chromatids. Okay, anaphase, what had happened, they have a deal. Okay, anti varanga addition. Okay, so what had happened, this two daughter chromatids split into each, okay, single chromosome, it divide into two daughter chromatids. Adu period. Okay, okay, then they move into this opposite pole. Okay, so they split into two and move into this opposite pole. It is in the anaphase. Telophase, main what it happens here, appearance of what, the nucleolus and nuclear membrane. So metaphase they disappear, in telophase they are appear in that one. Okay, another thing is here the spindle fiber. Okay, spindle fiber and astral rays, again what happened, disappear. When nuclear membrane disappear, and spindle fiber and this astral rays are disappear. Okay, and new endoplasmic reticulum is formed, Golgi complex also formed. Okay, so what had happened, telophase, appearance of nucleolus and nuclear membrane, and spindle fibers and astral rays are disappear, and new endoplasmic reticulum and Golgi complex are formed. Cytokinesis, I said it. Okay, two method, one is cell furrow and cell plate. Cell plate method, okay, what it happens, the vesicle, okay, cell plate, okay, vesicle, okay, cell plate, okay, vesicle, okay, so the vesicle, what it happened, okay, so vesicle provide by Golgi complex, okay, so this vesicles provided by Golgi apparatus, okay, so the unit of form a pharmacoplast, okay, so it form a pharmacoplast, okay, so pharmacoplast, okay, so it is the vesicle is provided by Golgi complex and form a pharmacoplast. And Furrow method, what it happens, okay, in Furrow method, what it is, cell furrow method, what it will happen, okay, the furrow appear in plasma membrane, PM, okay, plasma membrane. So here vesicle it's provided by Golgi apparatus and form pharmacoplast. Here the furrow appear in plasma membrane. This is the difference in this one, okay. It is furrow which is occur in animal cells, plate which is occur in plant cells, higher plant cells. So once again repeating, mitosis is the process, okay. So another name we called as mitosis also, it's also called equation division. Mitosis, another name we also called as equation divisions, another name of mitosis. So equation division, it's another name of mitosis. So first we have 5 stages: prophase, metaphase, anaphase, telophase and cytokinesis. So here the stage it is further, what are the process which occur? Prophase, short stage, thickening of chromosomes. Metaphase, equator plate it will be formed, disappearance of nucleolus and nuclear membrane and formation of spindle fiber. Anaphase, they are moved into this opposite pole and split into 2 daughter chromatids. Telophase, appearance of this nuclear and nuclear membrane and spindle fiber astrate to disappear and new endoplasmic reticulum and Golgi complex it is formed. Cytokinesis, it is formed in 2 stages. It's formed a vesicle, it provides the Golgi apparatus, furrow it is provided the plasma membrane, okay. So what is the significance when you say mitosis? What are the significance? Okay, why it is useful? Okay, what are the significance? What are the importance of this mitosis? Okay, mitosis important. Okay, significance or importance. Significance of this mitosis. Okay, mitosis mainly helpful for cell repair. Okay, mitosis very useful in cell repair. Okay, cell repair, the mitosis very important. And mitosis, it's production of diploid daughter cells. Okay, so mitosis, they can able to produce or production of this diploid daughter cells. Okay, and the daughter cell, whatever it is produced, they are identical. Okay, they are identical. Okay, and also genetically. Okay, they are identical genetically. Okay, genetically same. Okay, and fourth one is, okay, it is the mitosis which is helpful for restore nuclear and cytoplasmic ratio. So mitosis which is helpful for repair of this cell and it is produce the diploid daughter cell, and genetically daughter cell they will produce and restore this nuclear and cytoplasmic ratio. Fine. So it is helpful for this mitosis. So mitosis, when you want to more mitosis, okay, we want to do it. For that, it is we used auxin, cytokinin, okay, auxin, okay, cytokinin, gibberellin, okay. These are the components. It is called as mitogens. Mitogens. It is increase the mitotic division. Okay, this auxin, cytokinin, gibberellin, okay, and this are it's also okay, insulin, okay, insulin. This also it is increase the mitotic divisions more. Some it is called as okay, mitotic poison. Okay, this is a mitogen. This is okay, mitotic poison. Okay, some okay. What are the mitotic poison is compound? It is okay. Is chalons, colchicine. Okay, colchicine. Okay, this component it does affect this mitotic activity. Chalons and colchicine, it is affect the mitotic activity. That's the reason which is also called as mitotic poison. We call this. Okay, this is we have seen it. Next we move to this. Okay, so we have seen it. What is mitosis and who discovered it? Okay, and what are the process or stages in mitosis and what it is doing, and finally we have seen its significance and this what is inducing, what is inhibiting, we have seen it. Next we move to this meiosis. Okay, the meiosis, the process is discovered by okay, Farmer and Murray. Okay, so the meiosis it is they have occur in two, either it is occur in nuclear or cytoplasm. Okay, so it is occur in it is also occur in nuclear and cytoplasm. Okay, and finally it is they can single cell it able to produce 4 haploid cells. Okay, there it mitosis their diploid cell they can produce. Here they can produce okay, it is 4 haploid cell at the end of this meiosis divisions. Right now, the types of meiotic divisions. Okay, it is okay, either zygotic or it is spore or in the gamete. Okay, so the types of this zygotic after fertilization. Zygotic after fertilization. Spore after intermediate. Spore after intermediate. Gametic after okay, terminal meiosis. So types of meiotic division: zygotic after fertilization, spore after intermediate, gametic terminal after meiosis 1. Okay, so it is mainly occur in plants and algae and fungi. It is also happen. This is mainly zygotic is mainly fungi and protist. Okay, gametogenesis it is occur in plants and animals. It is occur. Okay, this is a meiosis 1. Now meiosis, it is also called meiotic or reduction division. Okay, also it's called as like equator division. It's also called mitosis. Meiosis also it is called as reduction and divisions. Okay, so now meiosis. Meiosis, I said, it is 1 and 2. Okay, we have about 4 stages: prophase, 1 metaphase, 1 anaphase, 1 telophase, 1. Like metaphase, okay, pro 2, meta phase 2, anaph 2, and telo 2. So like it is the process which is occur in the meiosis. Okay, and the prophase further divided into 5 category. Okay, leptotin. Okay, leptotin. Okay, leptotin. Okay, prophase. Zygotin. Okay, packeting. Okay, diplotin. Next, the final one, diakinensis. These are the 5 stages which is occur in prophase 1 alone. Okay, it is a long and complex process. Okay, very long. Okay, the meiosis very long and lengthy process which is occur in the prophase 1. Okay, right? The leptotin. Okay, so these are the 5 stages. Okay, leptotin. If one person, for you your understanding, I am selling simple. Okay, ஒருத்தனுக்கு leprosy வந்துச்சு, அதனால சைக்கோ வானா, அவனை சைக்கோனால பேக் பண்ணலாம், டிப் பண்ணல அவன் டயாவா மாறிட்டான். leprosy, psycho, pack பண்றோம், dip பண்றோம், டயாவா மாறுறோம். Okay, leptotin, psychotin, picotin, and diplotin and diakinesis. Okay, these are the process which is occur in the meiosis 1. Okay, so meiosis 1, especially in the prophase. Right. Here you need to know that process, what are the process and what are the function it happens. Okay, அப்படிங்கறது நீங்க தெரிஞ்சுக்கோங்க. Okay, here mainly astral rays are developed. Astral rays are developed. Okay, at the centriole start moving. Okay, centriole start, astral rays develop, centriole start moving. Okay, here then second thing is psychotin, this process which is occur. Okay, bivalent chromosomes. Okay, bivalent chromosomes it is formed. And another thing is synapses. Okay, synaptical complex. Synaptical complex. Okay, bivalent chromosomes, synapses, and synaptical complex which is occur in this one. Okay, packeting, it is also formed. Okay, sister and non-sister chromatids. And main thing is another crossing over. Crossing over incident is occur in packeting. Okay, crossing over it is mainly. Okay, and this packeting, it is mainly non-sister chromatids are formed. And one more thing is packeting also, it is one more thing is it is called recombination. Okay, recombination. Okay, so or else it's called crossing over. Diplotin, it is among this leptotin, diplotin, psychotin, it's the longest stage in this meiosis 1. Okay, longest stage in meiosis 1, diplotin, it's the longest phase. And here what is happened, desynapsis. Okay, synapses which is occur in this cytokine. Diplotin, they have a D synapses, and also it is the longest one. And this the area which is the cell, it form X shaped. The cell which is formed X shaped in structure. Okay, X shaped in structure. And diakinesis, what will happen? It is a terminalization. It's go to the terminal. Okay, first acetyl day developed, centrioliters start moving, and cytokine bivalent synapses synaptical complex packaging. Okay, it is sister non-sister chromatids are formed, crossing over it is occur, recombination it is occur, and diplotin mainly it is the longest stage which is occur in prophase 1, and this among this leptotin, diplotin, and cytokine and diakinesis. So D synapses is formed, and the chromosome which is formed X shaped in structure. Terminalization it is also occur in the diakinesis. Now, so we can move into this. This is only occur in what? Prophase 1. Okay, now we move this. What it occur in metaphase 1 and this anaphase 1 and telophase 1. Okay, now it is metaphase 1. Anaphase 1. Telophase 1. Okay, but meiosis 2. Prophase 2. Metaphase 2. Anaphase 2. Telophase 2. Okay, now இங்க என்னென்ன process metaphase 1 ல என்ன நடக்குதுன்னா, okay, it is formed a bivalent stage. Bivalent stage. Okay, so bivalent stage, and also it is okay, it has microtubules. Okay, microtubules. So it is formed. Okay, bivalent stage and microtubules are formed. Anaphase 1 is here. Main thing is disjunction. It is junction chromosome disjunction of this one. Okay, and also dyad. So the word is very important. Okay, crossing over, and synapses, desynapses like disjunction, dyad, which is formed in anaphase 1. Dyad. Okay, dyad. Dyad means the half number of chromosome. Half number of chromosome. Half number of chromosomes. Dyad, which is talk about half number of chromosomes. Okay, bivalent stage just occurred in metaphase 1, and this anaphase 1 it is disjunctions or dyad. Dyad means half number of chromosome which is occur 1. And telophase, what will happen? Okay, and this uncoiled and elongated the chromosome. Uncoiled and elongated the chromosomes. Okay, now in between they have a cytokinesis. Okay, so next cytokinesis mainly, okay, the cytoplasm. Okay, what happens? Cytokinesis, cytoplasm, what? Okay, and other organelles. Okay, so other organelles, what will happen? They are equal in number. So diakinesis, cytoplasm and other organelles, it is form equal in numbers. Okay, next meiosis 2. Okay, so it is also called okay homotypic division. So bivalent stage, disjunction, dyad, it happens in this. Prophase mainly duplications or separation. duplication or separation, okay, separation of pair of chromosome. Okay, and metaphase, what will happen? Okay, align equator, align the equator regions. Okay, so anaphase, centriole start to divide. Centriole start to divide. Telophase, okay, so it appear nuclear, nucleolus and nuclear membrane appear. So these are the stage which is occur in this meiosis 1. Okay, these are the stages which is occur in 1. Okay, meiosis. Meiosis mainly helpful for evaluationary. Okay, so it is mainly helpful for evaluationary analysis. So it's meiosis the process which is helpful for evaluationary analysis of this number cell. Now we move to these questions. So, நீங்க question comment box ல சொல்லலாமா? Question comment box. Okay, நான் questions இது பண்றேன். நீங்க பார்த்துட்டு answer your நீங்க என்ன பண்ணலாம் comment box ல இது பண்ணலாம். Okay. The complex, okay, the complex form, yeah, a complex formed here a complex formed by a synapsed pair of synapsed pair of synapsed homologous chromosomes is called A. Kinetochore, B. Bivalent, C. Axonym, D. Equatorial plate. Okay, the complex, okay, formed by pair of synapsed homologous chromosome called, okay, either bivalent or kinetochore or axonym, okay, or else equatorial plate. You can type your answer in the comment box. You can type your answer in the comment box. Answer Yeah, thank you Nitya and Madhu. Yeah, B is right. Yeah, right. Yeah, meiosis 1 it has happened. Okay. Yeah, B, it's right. Thank you Santosh, Priya. Okay, so it is the synapses, it is also called bivalent. Okay, bivalent, it is homologous chromosome, it joined together, it is mainly occur meiosis 1. Okay, meiosis 1, I said it is okay leptotin, and after that it is again zygote. Okay, zygote stage mainly it is occur this process. Okay, especially zygote stage, this is mainly it is occur bivalent stage. Okay, next. Which one of the following characteristic, okay, which one of the following characteristics, okay, feature during meiosis? Okay, which one of the following characteristics feature meiosis in somatic cell? Option A, spindle fibers. Option B, disappearance of nucleus. C, chromosome movement. D, synapses. You can also type your answers. Okay, so which one of the following characteristics features during meiosis in somatic cell? Option A, spindle fiber, disappearance of chromosomes, and chromosome movement and synapses. You can type your answer, either spindle fiber, or it is synapses, or it is disappearance of chromosomes. Answer, Dinesh, Santosh, Priya. Yeah, you can type your answers. C. Thank you, Kavita. Thank you. Synapses. Yeah, win. Okay, right. Thank you. Synapses is answer. It is occur main meiosis. Okay, they are asking meiosis. So spindle fiber formation, it is mitosis also occur. Disappearance of chromosome, movement of chromosome, it is also occur in mitosis also. The main synapses, the process. Synapses, it is a homologous chromosome that it is formed. Okay, homologous non-sister chromatids. Okay, it is formed. Two homologous chromosomes. Okay, it non-sister chromatids formed synapses. Okay, this is a main things it has happened. The synapses, it is also formed during the time of zygote. Okay, synapses, it is formed in the stage of zygote. It is homologous chromosome formed together. Okay, thank you. Okay, it's not B, its answer is D. Okay, okay, synapses. Next questions. During metaphase stage of mitosis, spindle fiber, okay, during metaphase stage of mitosis, spindle fiber attached to chromosomes at, okay, so option A, centromere, B, kinetochore, C, both A and B, C, centromere and kinetochore, okay, and areas adjoining centromere. Okay. Okay. So first centromere, kinetochore, both A and B. Centromere, kinetochore, and were area of adjoining centromere. Okay. Area of adjoining. The last one, area adjoining centromere. Answer. Yeah. Thank you, Kavita. Thank you, Nitya. Yeah, thank you, Priya, Santosh. Okay, and Santosh, yeah, the answer is kinetochore. Yeah, we are right. Okay, so the answer is kinetochore. The kinetochore, it is mainly used. Okay, the kinetochore. B is the answer. It's a kinetochore. It is mainly the one which is self. Whatever you said, it right. Okay, kinetochore. It is a small. Mainly this is small disc shaped one. Okay, the kinetochore, which is okay, small and disc shaped structure, which is attached to the centromere, okay, this spindle fiber or the chromosomes. Okay, so it is the cell, it is go to center in position. So the kinetochore, it is helping us to cell move into the center one. Okay, next. If a diploid cell, okay, if a diploid cell is treated with Colchin, okay, Colchin, okay, Colchin, then it becomes A triploid, B tetraploid, C diploid, D monoploid. Okay. So if a diploid cell treated with Colchin, then the cell become a triploid or tetraploid or diploid or monoploid. You can type your answers. You can type your answers. Answer Dinesh B. Okay. Next. Nitya. Okay. Dinesh, Nitya, and Dinesh, Kavita. Thank you. Madhu. Thank you. Yeah, you are right. Thank you. So answer is tetraploid. Okay, because this is a alkaloid. Okay, this is alkaloid which is inhibit the growth of spindle fibers. Okay. So this colchicine is alkaloid. This inhibit this spindle fiber formation. That's the reason it has become a tetraploid. This cell. Okay. All right. Thank you. So thank you for your listening and watching this program. Continuously be with us and watch over this program. So we'll meet you tomorrow with biomolecule. Okay. If you have any doubt, you can contact us. Okay, through over the phone or mail. So please register us as soon as possible. Thank you so much.